So chapter 12 is about mitosis, and you guys are probably very familiar with this stuff already, so we're just going to kind of zoom through it. So mitosis is cell division, right? So the ability of organisms to produce more of their own kind best distinguishes living things from non-living things. So we can make more. Humans can make more humans, dogs can make more dogs, cells can make more cells, and this is how cells make more cells. Those are the chromosomes, the DNA of a cell as it's dividing. When we're done with this, you should be able to identify all the different stages and what's happening here. That's actually a very cool picture. So, in single-celled organisms, cell division is what makes a whole new organism, right? And so, multicellular, because only one cell. Take one cell, divide it in two, now you have two. You reproduce. Whereas in multicellular organisms, like humans, we have development from a fertilized cell, growth, and repair. So we have really big life cycle. That's pretty obvious. But cell division is really important part of the cell cycle, which is like the life of a cell from when it's formed to when it divides again. So we're going to talk about the cell cycle. So most cell division results in genetically identical daughter cells. So that's really important. Mitosis gives you two genetically identical, they are exactly the same genetically, daughter cells. So we call them daughter cells after we divide, and they are identical, and there's two of them. The only exception to this is meiosis, which makes gametes and such sperm or egg cells, and you get 4 daughter cells, and they are non-identical in meiosis. But mitosis gives us 2 identical daughter cells. So let's talk about DNA. So all of the DNA inside of a cell, we call the cell's genome. So it's all the DNA. So if you guys have ever heard of like mapping the human genome, we now know where every single gene is in all of our 46 chromosomes. And so a genome can consist of one DNA molecule. If we're talking about a bacterial cell or another prokaryote, they only have one DNA molecule, whereas we have 46 different DNA molecules. So the genome can consist of many different kinds of DNA. And inside a cell, our DNA is packaged into what we call chromosomes. So those are, that is DNA inside of a cell. What, is there anything in particular? That is a eukaryotic cell. You can see the chromosomes, so that means that we know it's preparing to divide. So when we're not dividing, our cells, our DNA is stored in what's called chromatin. And chromatin is basically like, it looks like a cup of noodles. Each noodle is a long DNA strand, and they're all just there. And that's what it's like during most of the cell cycle, when your cell is just going along doing its job. And we have, like I said, 2 kinds of cells. We have all of the cells inside your body, which are the somatic cells, and then we have our reproductive cells, which are our gametes. So somatic cells and gametes. Somatic cells do mitosis, gametes do meiosis. Okay, so during the cell cycle, we have to replicate our DNA, because how else are we going to get an even number into both of them, right? So each cell has the perfect amount of DNA, but to make 2 new cells, we have to double that. That way that we have 1 set for one cell, 1 set for another. And the chromosomes condense. What that means is it goes from looking like a bowl of noodles into those big X-shaped things that we are familiar with. So that's what chromosomes condensing is. And so this is what you've probably referred to as a chromosome, this X. And each chromosome, once it's condensed, is made up of 2 sister chromatids, and they're identical. So this is one sister chromatid, this is another sister chromatid. They are exactly the same, okay? So chromatin is a bowl of noodles, chromosome is the X, chromatid is one of these. And the centromere is where they attach. So this is a good picture. So here's, the whole thing is a chromosome. It's very tightly condensed. So all of this is all those noodles packed super, super, super tightly together. And this whole thing is a chromosome. This is the centromere where they attach, like where the 2 lines cross in the X, and one of these is a sister chromatid. One sister chromatid, second sister chromatid. So during cell division, what we're going to do is we're going to pull these sister chromatids apart, and now as soon as they separate, they become chromosomes. So this is one chromosome, sister chromatid, they're attached to the centromere. We pull them apart during division, and now I have two chromosomes. So as soon as they separate, they're called chromosomes again. There's a good image of that if you need that. So the cell division consists of two parts, which might be different than what you have thought before. You probably thought cytokinesis was the last step of cell division. Or last step of mitosis is actually different. So mitosis is the division of the nucleus, and cytokinesis is division of the cytoplasm. So they happen simultaneously, but they're actually little different processes, and we'll talk about that. Okay, so on to 10.2. This is when we're getting into the nitty-gritty of this. Don't worry about the experiments. So we have two stages of the whole cell cycle. There's interphase, which is when everything is happening inside the cell. The cell is just out there being a cell. If it's an eye cell, it's seeing. If it's a heart cell, it's pumping blood. If it's a blood cell, it's carrying oxygen. So it's doing its job. It's growing, it's developing, it's doing its job. But then it needs to divide. So it goes into M phase, which is mitosis, the mitotic phase. But interphase is made up of three different stages called G1, S, and G2. The G stands for gap, but we can call it growth because they're also growing. And the chromosomes are replicated during the S phase, which is very important. That's when our DNA replication happens. So here's a picture of the cell cycle. We start with one cell, goes through its whole entire life. You can see interphase is the grand majority of the cell cycle. G1 is the biggest part of interphase. Then during S phase, we replicate the DNA. We make a whole new set of DNA. And then during G2, we get ready for mitosis. And then mitosis and cytokinesis happen during mitotic phase, during M phase. And so we have 5 phases. Prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis will overlap with telophase. So here's a picture. Okay. So this is what the cell looks like during the end of interphase, during G2. So we have the chromosomes are in chromatin. They are not condensed. They are contained by the nuclear envelope inside of the nucleus. You can still see the nucleolus. And then we have these things called centrioles. Centrioles are microtubule organizing centers. So basically, we're going to use microtubules, which are a part of the cytoskeleton, a lot for cell division. It's very, very important. And so this whole thing is the centrosome, and then these little guys are the centrioles. So that's what it looks like. Oh, so we have these two centrioles, and they make the microtubules. That's where the microtubules get made. We're going to call microtubules mitotic spindle during mitosis. So they are just microtubules. Remember, microtubules are really long fibers, and they are made up of tubulin is the monomer. So we can have 5 tubulin monomers, or we can have 5 million tubulin monomers, making a really long microtubule. So that is G2 of interphase, just regular looking cell. But remember that this is we have 2 copies of the DNA in here. We've already duplicated it. Then we move into M phase, we move into prophase. The important events during prophase is that now the DNA is no longer stored in chromatin, it's stored in chromosomes. You can actually see the X's. It has condensed into these structures. The nuclear envelope is still in place. You can see that here. It hasn't broken apart yet, but the centrioles have started to move away from each other. You can see they're starting to move to opposite ends of the cell. And they start producing these long strands of microtubules out of them. And those are called asters. So we have a centriole, and sticking out everywhere around the centriole is a bunch of different microtubules. And those are, they form a radial array, and that's called the asters. So that's prophase. The main events during prophase is we start to see the chromosomes, centrioles start to separate. Then we get to prometaphase. This is not even always classified as a step, but what's happening here is the centrioles have completely moved to opposite ends of the cell, but they have mitotic spindle reaching from one end to the other. Now, the nuclear envelope is gone. it's disintegrated. It's not there anymore. And so now the chromosomes are free inside the cell. And what happens is the mitotic spindle reaches out and attaches to the centromere of the chromosomes. So you can see that. They are just floating around inside the cell, and the mitotic spindle has reached out and attached to the centromere. There are also many microtubules that are not attached to the kinetochores of the chromosomes. Now, the difference between kinetochore and centromere is that centromere is a location on the chromosome, so it's just this place in the middle, whereas the kinetochore is a bunch of proteins that are used to help attach the chromosomes to the mitotic spindle. Okay, then we get to metaphase. So the purpose of attaching the microtubules to the chromosomes is we are going to pull them into a line called the metaphase plate. So you can see all of the chromosomes are perfectly in a line. So these there's a microtubule from each centriole attached to the kinetochore, and so they pull each other back and forth until they line up in a straight line. It's like two fishing poles attached to one chromosome, and they pull them into a straight line. That's metaphase. No nuclear envelope, centrioles are at opposite ends of the cells, etc. Then what happens is, during anaphase, the cell starts reeling in its mitotic spindle. Starts breaking off tubulin monomers and making it smaller, okay? And as that happens, the two sister chromatids get yanked apart. The kinetochore breaks in half, the centromere, they split at the centromere, and then they're pulled apart. Now they are, again, chromosomes. They're not sister chromatids anymore, they are chromosomes. And they get pulled to opposite ends of the cell by the mitotic spindle. As this is happening, the cell is elongating, so you can see that it's starting to get pulled apart. Then we get into telophase and cytokinesis, and these happen simultaneously, but remember, cytokinesis is division of the cytoplasm, and mitosis is division of the nucleus. So what's happening with the nucleus during telophase is that the nuclear envelope is starting to reform, and the chromosomes are starting to condense, and the nucleolus is starting to reform. So the nucleolus was gone during this whole thing, but it's starting to reform again now. And now you can see each cell has a nucleus and a centriole. So we're done. So now we just need to split the cytoplasm. So that's what happens during cytokinesis. So basically, it's like we have this balloon. Someone ties a string around the middle of the balloon and starts just tighten and tighten and tighten the string. The balloon's going to form two little balloons until eventually it would pop into two if you pull it tight enough. And that's what happens here. We have an actin filament. So remember that actin filaments are microfilaments, another kind of cytoskeleton. And what it does is it pulls and pulls and pulls, just like tightens a ring. It's called the contractile ring around the cell until the cytoplasm divides and we have two daughter cells. These are some great images of mitosis. So you can see during G2 of interphase, this blue stuff's the DNA. It's not condensed. The green stuff is the microtubules, and so they're just kind of everywhere. And then you can see this is the centromere. I'm sorry, the centrosome. So that's where all the microtubules are coming out of. It's a microtubule organizing center. But you can see that they're spreading out everywhere. And when you look, you can start to see the chromosomes condensing. It's not just a blue mass. You can actually see them condensing. Then during prometaphase, you can see that they've separated out. The nuclear envelope is gone. They're not just stuck in here anymore. And the centrosomes are at opposite ends of the cell. They've moved, and now they're starting to spread out and attach to the chromosomes. Then during metaphase, you can see all of the mitotic spindle. They pulled all of the chromosomes to the center. That's not the best picture I've ever seen of metaphase, but you can see they're all in one spot in the cell. Then during anaphase, the centrosomes start to pull apart, start reeling in their mitotic spindle, and they split the sister chromatids at the centromere. Then during telophase and cytokinesis, we can see that we have pulled our DNA all the way to one end of the cell, and it's starting to recondense or uncondense, and the cytoplasm is starting to divide.